Good morning, everyone, and a very warm welcome to the Barnett Lecture 2020. My name is Ben Swallow, and I'm a member of the Conference Board and an officer on the Environmental Statistics Section Committee. I'm delighted to be able to welcome you to this virtual Barnett Lecture delivered by this year's recipient of the Barnett Award. For those of you who may not be aware, the Barnett Award is an honour bestowed by the RSS annually on a statistician who has made a considerable contribution to the field of environmental statistics, which also incorporates environmental health and ecological statistics. And it's for his contribution to the field of ecological statistics that this year's recipient has been awarded this honour. As well as being a fellow member of the Welsh Statistical Ecology contingent, he has received many accolades and achievements over the duration of his career that I don't have time to list in full today, but they do include being the first director of the National Centre for Statistical Ecology, as well as a joint founder of the now biennial International Statistical Ecology Conference that has grown significantly over the last few years and seen its biggest attendance this year. He has made a significant contribution to both methodological literature across statistical ecology and consolidated the UK as a global leader in the field, as well as significant contributions to applied ecology literature. He's a leading authority on age-structured modelling of ring recovery data, and was one of the first people to propose the inclusion of weather covariates in modelling survival probabilities. He's also a leading founder of the subfield of integrated population modelling, combining data sources to estimate both abundance and demographic parameters. And he has been an inspiring mentor for many junior academics, PhD students and postdocs, as well as an inspiring collaborator to both statisticians and ecologists alike. It is therefore with great pleasure that I introduce the recipient of this year's Barnett Award to deliver the virtual Barnett Lecture 2020, Professor Byron Morgan. Thank you, Byron.
Hey, thank you very much. And thank you, Ben, for the very kind introduction. Here is the title of the talk. And I would like to start by acknowledging my many collaborators who've we've worked on papers together in this area, and we are continuing to do so. Emily Dennis is at the top of this list here because she got me into the area of butterfly modeling, and several of the examples I will give are hers. Okay. We are just coming to the end of a decade of the UN Decade of Biodiversity. And in her Guy Bronze Medal speech yesterday, Rachel McRae commented on the declines in biodiversity throughout the world that we're all concerned about. But how do we know? How do we know what's going on? Are things really going down? Are they going up? How serious is it? Now, one way of looking at this is through things called multi-species indicators, where we have, over time, basically a graph for a group of species. And we can look at that and see how it's performing. So the idea behind this talk this morning is to lift the lid, really, on multi-species indicators and look at what goes on underneath and how they're formed. What we've been doing is coming up with new methods, trying to improve them and trying to make them faster, more efficient. So we're going to look at indicators for abundance, how many things there are, and distribution, where things are. This requires probability models, and we'll look at some simple models for abundance and occupancy. We'll give an example of functional principal component analysis, where we can look at individual species indices to see whether there's too much variation, really, for these to be combined in an indicator. And then we'll finish off with a new indicator that several of us are involved in, which has just been published. So that's the outline for the talk. There are many examples of multi-species indicators. And here is just an example: the state of nature, the Living Planet Index, state of UK birds, and here's a Welsh one as well. There are many examples of multi-species indicators. Here they are. And I'll look at illustrations from the state of the UK birds, but primarily we're going to talk about butterflies. Both of these reports result from really massive surveys. Here are two in the UK birds report, for example, and there's the UK Butterfly Monitoring Scheme survey, which has been running for some 44 years. It runs for half the year with people going out each week and counting butterflies. And there's also Butterflies for the New Millennium, which records citizen science, more amateur-like collections of data. So looking at bird indicators, here are three examples: farmland birds, woodland birds, and wetland birds. So what's being done here then is that we don't have all birds. Birds are divided up into similar groups. And within each group then, what we have here is an indicator. Each year, a model is used to produce an index for each species, okay? And then, sorry, can I check? Can you hear me? Yeah, okay. And then these are combined to form an indicator. So this is a succinct summary of a very complex system, but it's what people need. And importantly, these indicators inform government policy. Of course, there's information lost, and you can get some idea of what is lost through bar plots for different periods, for the long term and for the short term, say over the last 10 years. Okay. Here are some more multi-species indicators, and these are for the UK monitoring scheme data. So the black dots here then are for all butterflies, and they've been divided up to give indicators also for specialist butterflies that you only find in particular places, and much more common butterflies that you find in the wider countryside. So these are indicators then for abundance, and a geometric average is being used here so that for any year t, this is what is done, where NIT is the count of the number of individuals of species I at that time. Okay. So that's for abundance. And here then is another one for distribution, okay, using the butterflies of the new millennium. And you can see it's a slightly different picture, or somewhat different picture from before, with the specialist butterflies really declining in terms of distribution, and the common butterflies not really declining. And this is the overall picture reflecting what's going on down here. Okay, here's a recent analysis of Scottish moths, just to emphasize the point that abundance and distribution are different. So what we have here is the geometric multi-species indicator for counts, which is going down, whereas the occupancy indicator is going up. So these things aren't the same, and they require different models. So moving on to models, and from now I'm just going to focus on butterflies. There are all sorts of aspects to modeling. You can have static models that analyze data 1 year at a time, or you can have dynamic models that analyze the complete set of data all at once. And we'll see some examples of both of these. what we have in the panels on the right here are data from just 2 sites in just 1 year. And until recently, people used GAMs to produce a smoothing of the data, the counts being represented then by these crosses here. These are 3 different butterfly species, and they reflect the kind of variety of shapes that models have to describe. Chalk hill blues are univoltine. There's just 1 period in the year when they're on the wing, the adults are flying. The Adonis blues pop up twice a year, and speckled woods are multivoltine, and they are more generally seen throughout the year. This is the UK situation. You go south into Europe, and the picture will change. So up until recently, people have used generalized additive models to smooth the data before going on to form multi-species indicators, but it's very slow. And you know, if for butterflies in the UK, we have 59 species. If analyzing each one each year is slow, it's a problem for producing these annual multi-species indicators. So the challenges then are how to account for seasonal variation in a model. There's missing data for the citizen science data, the distribution data. We have over 40,000 sites. There's a lot of information there. And as we've just seen, there's variation between species. So what we've set out to do is to come up with methods that can be sufficiently flexible that they can be applied to many different species. Okay, and we've tried to make things faster. And as well as producing multi-species indicators, we've also tried to understand better what is going on, suggesting new avenues for research. So here is the first model. It's a generalized abundance index, and this is for counts. Okay, so for any species and any year, the model assumes that we can describe the counts. Here they are, visit J to site I, account Yij. We just have a product then of, if it's a Poisson model, of these Poisson probabilities. And the Poisson means are a product of the abundance at site I, at this time for this species, times a shape. And it is this shape then that we design for butterflies. Now, if you maximize this likelihood, conveniently, you can get this estimate for the ith site for the abundance on the site. Okay, and you can see, excuse me, that it's just a scaling of a site total. Okay, so this shape term can be, is a function of the other model parameters. So if we substitute this back into the likelihood, then we greatly reduce the dimensionality of the likelihood, the sample space. The parameter space is hugely reduced in size, and it makes it much easier to maximize. Once we've got the maximum likelihood estimates for the parameters theta, then we can calculate these things, and we have estimates for all of the model parameters. So this approach, it works conveniently for these models. It's called concentrated likelihood or profile likelihood, and it makes things much faster. And it applies also to more complicated models, say with a negative binomial or zero inflated, rather than just the simple Poisson. And in addition, we'll bring in covariates, trying to describe key parameters in ways that will improve understanding. Okay, about the shape. Well, for butterflies, we have 3 possibilities. We can put a spline in there, so this is like a GAM. We can put in a mixture. We've used a mixture of normal densities to reflect the shapes that we saw on an earlier slide, or we can use a stopover model, which is more complex, and it involves these really these entrance probabilities. These are designed to produce the necessary shapes, and they tell us when adult butterflies start to fly. And then when we see them, from the start to when we see them, there has to be a product of survival probabilities for the butterflies, and that's what we get here. So this is a complex model. I'm not going to show results from this, but it actually gives us a way of estimating butterfly survival, about which not much is known. Okay, so here are some results from doing this for 6 species of butterfly. Okay, and what's in red here is what we get from the GAM approach, okay, for comparison with what we get from using, in fact, a mixture component of the generalized additive index approach. And you can see that these things are fairly similar in shape. And for the Poisson component of the GAI model, the speed is 75 times faster than using the GAM. This approach, due to its speed and flexibility, is now widely used. So, for example, each year there is a report on the UK Butterfly Monitoring Scheme results, and it uses this model. It's now used for modeling European butterflies in an organization that does that. And last year, the first ever atlas of large moths in Britain and Northern Ireland, I think, used this approach. Here's an illustration of the value of being able to have covariates in a model, looking at the timing of events, or phenology as it's called. And this is for just 1 year, fitting a mixture generalized additive model, a GAI model to common blue abundance data. And the two means, assuming a bivoltine species, and the mixture probability are regressed on northing, telling you how far north you are. So what we find here, and this was found several years ago in a paper, though Emily produced this graph just a couple of weeks ago, that in the south, so I'm just about here, for example, in the warm south, where it's probably going to be 25 degrees today, butterflies have enough time to squeeze in 2 periods when the adults are flying. Okay, and after the first period, then the rest of the life cycle takes place before the second time when the butterflies are on the wing occurs. But as you move further north, the peaks come together until you get where Ben is in Scotland, and you find there's just the 1 period when the adults are on the wing. Now, this is just for 1 year. If you do this for different years, then the latitude, the northing at which you lose the bivoltine aspect, switches and moves further north due to global heating. Right, those were all static models, analyzing data one at a time. For dynamic models, we link the years together through an idea of a productivity parameter. So here is a year for a bivoltine species. Here's another year. And the first period feeds through into the second period. And then the second period in 1 year feeds through into the first period in the following year. So this way, we link the years together. The model is basically a hidden Markov model. And as a result of doing this, and this is for for various Univoltine species, in the panels in the left-hand column, we are comparing the simplistic, slow GAM approach 1 year at a time. These indices are compared with what we get from the dynamic model that I've just mentioned. So there's good agreement here. But in addition, now we get these estimates of productivity, varying with time here. And this is a new way of looking at these data. So we can go to ecologists and say, what do you make of this? Here is a gatekeeper, which is declining, and here is its productivity, whereas here is a marbled white, which is increasing in numbers. Here is its productivity. What do you make of this? And in terms of bivoltine species, here are 5 of those. The GAM approach would give you just a single index. Okay, that's what we get in red. But as we have these 2 periods of flying, we can now construct indices for the, indices for the first brood and indices for the second brood separately. So this is, again, it's more informative. And we get these measures then of productivity for the 2 broods. Okay, once you've got a model with covariates in, you can see what the model will tell you. Going back to Univertan examples here, these 6 Univertan examples. Here we've looked at modeling productivity as a function of how far north you are and minimum winter temperature. And apart from the gatekeeper down here, in all of these cases, as the winter gets colder, then the productivity goes up. And also the further north you are, the more productive these butterflies are compared with the ones further south, the ones in red. That might seem a strange finding, but it's thought that cold weather is actually good for the overwintering stages of butterflies by killing off parasites, for example, and giving butterflies a better start in life. Okay, moving on now then to occupancy models. These are different types of models, and they've been incredibly important and influential in ecology generally over the last 20 years. They're very simple. And what we have here is information now then on a species rather than information on individuals. So for any species, there are records made at various sites, surveyed a number of times within a fixed season. And what we get then is a data vector for the ith site, which in principle is a set of zeros and ones. Ones meaning the species was there, and zeros meaning the species wasn't there. We sometimes don't have zeros in these records, and then they have to be imputed from information on other species that were seen at the sites at the same time. So when you have an encounter history of zeros and ones at the ith site, and here we have an indicator variable telling us whether the site is occupied or not, for the year in question, so Zi equals 1 means the species is there, then we just have this simple product of Bernoulli terms to be the probability of the data conditional on a species being occupied. So this important term here, the detection probability, can be a function of covariates. But what we are really interested in is whether the site is occupied or not. And the likelihood then has occupancy parameters, which can vary with time and site and so forth. And what we had on the last slide is multiplied by the probability that Zi is 1, that the site is occupied. And then we have this additional term to describe a complete set of zeros for the y vector. Okay. So you can get zeros when a site is occupied. You just didn't see the species. But you can also get zeros by the site not being occupied. And here again, this is a key model parameter, and we can write it as a function of covariates. This is just an illustration for the large skipper butterfly, where the index for this species is described in terms of northing, how far north sites are. And this is useful, showing where a species is doing well, where it's not so not doing so well. And in spatial terms, then, this is just for 1 year. These are the data where the sites where this species, the large skipper, again was seen. These are the index values, the average occupancies for the sites, and these are the estimated standard errors to help us understand how precise these fitted values are. This is just for 1 year, and you can do this for each year, and then produce a movie. And Emily has done that. So if a species is on the move, for example, then that might become apparent from the movie. Okay, I've just been talking about data from the butterflies for the New Millennium database, and this is all to do with species and occupancy. And what has gone into the analysis that I'm going to be talking about is what we have here for 48 butterfly species. Okay, so what we have here now then are estimates of occupancy, giving us an index for each of these 48 species. Now, each square involves fitting a model for each year. And if you do the sum, 1,680 models, therefore, are fitted to produce these plots here, these indices of occupancy. So you can see that some of these species are declining in terms of occupancy, whereas others are doing really very well in terms of occupancy. So just throwing all these together, these indices together to form a multi-species indicator, may not be a great idea. So one possibility is just to use functional principal components. It's a simple way for looking at structure in a set of index curves. And this is the result of doing this for butterflies for the New Millennium data that were illustrated in the last slide. And we can use these harmonic plots in a standard way to interpret the principal component functions that come out. And the first component here, which describes most of the variation between the index plots, is just a measure of the butterflies that are the species that are seen in many places, as opposed to the species that aren't seen in many places. Okay, that's a standard sort of first component from principal components. And as is often the case, the second component is possibly more interesting, and it distinguishes between the species that are declining in terms of occupancy, and those that are increasing in terms of occupancy. And this is found also from a much larger data set, the 230 species of Scottish moths, with more variation here for the second component. The second component is basically a measure of trend. And for each of these species, plotting trend along here and the score on the second component up there, we get not a straight line, but something that shows that we're measuring trend through that second species. Now, you probably won't know much about butterflies, but if you do, this is a wall butterfly. That's a small heath. Purple hairstreak and so forth. These are up here because their trends are going down, whereas speckled wood, holly blue, Essex skipper, and so on are going up. And if you do the first two component plot of the scores on those components for these butterflies, then this is what we get. Now here, the butterflies are divided up or color-coded really, primarily in terms of whether they are habitat specialists or seen more widely. So we get the wider countryside ones over here, the meadow brown, for example, a brown butterfly that's more or less just come to the end of its flight period, but is pretty much all over the place. And over here, you get a species like the chalk-hill blue, high brown fritillary, Duke of Burgundy, and so on, that you only find in very singular special places. Kent, for example, is famous for its chalk downland, its beautiful flower meadows, and it's one of the very few places in Britain that you can see the Duke of Burgundy. So this kind of check of indices is useful to see whether it would be an idea to divide your data set up to do multi-species indicators for subsets. And of course, that is what is done for the birds, the farmland birds, and so on, that we saw at the start of the talk. And it's what is done naturally for people who understand their butterflies. Okay, so here are, going back to the count survey, here are two indicators, two illustrations. And in fact, there are two indicators in each of these panels, one for specialists and one for the wider countryside species. And these are very similar to what we saw earlier, except that these indicators aren't formed using a geometric average. They're formed using this new approach, which I'm now going to describe in the last few minutes of the talk. Okay, so this new indicator does things that other indicators don't do. And in particular then, as this is just an overview, I'll explain how it deals with gaps in the records, and in particular with species that enter records late. Not all species enter time periods at the same time periods of survey. Okay, so for example, for the butterfly monitoring scheme, when it started in '76, there weren't that many sites, and only 36 species were recorded. And then in succeeding years, as the survey became larger, more species were added to the list. So in 1977, there was an extra one, and 79, there were 9 extra ones, and so forth. So we need to have a way of dealing with this. And the current way is not as good, we think, as what we have in the new method. So the new model then is this. So what we count is denoted by Y here, and there's a lot of assumptions of normal distributions here, and that can be changed. The counts are supposed to be normally distributed about a mean, which is an annual index for a site S at time t. Okay, now this, of course, is hidden. And these R's here, so these R's here, are growth rates. So they reflect the changes in the annual index that we don't see from year to year. And what is at the basis of this multi-species indicator is having these R's for different species sharing the same distribution with a multi-species average growth term, G. And that is how this model, based on growth rates, provides a multi-species indicator. It produces these G's, terms in fact that we can smooth. Now, this is a state space model, and the state space model formulation excludes the growth rates. You can get rid of them, and this is what you're left with. Okay, this is what you observe. That's the observation equation. And then the state space equations, you need an initialization assumption. And then after that, this is what you get. The next annual index for this species is normally distributed about the last one, added on to the multi-species average growth. Now, as it's formulated here, this is a linear Gaussian model, and it's then very easily fitted and very efficiently fitted. Okay, so this is the state space formulation, and by analogy with you get these R's from the S's, so there's a recursion for the R's, okay, which I haven't got on this slide, and that recursion motivates what is happening here. So this term here, by adding up the average, the G's were the multi-species average growth values for any time. By adding these up, we get something that we can plot as a multi-species indicator. We can smooth it and so forth. It's a Gompert state space model, easily fitted by the Kalman filter, which also deals very easily with missing values. The whole thing can be made much more general in a hidden Markov modeling framework, which also deals really transparently and easily with missing information. We don't have to make assumptions for dealing with missing data. Okay, and this can be applied to occupancy data. It can be as well as to abundance data. And it's fast. Like everything I've talked about, I've been using classical statistics, but we have a Bayesian implementation of this as well. So to conclude, here it is, this new method applied to bats. We have 17 species of bat in the UK, but only data providing indices for 8 species. And down here then is the multi-species indicator. There are 2 there, and there's an extra one that I haven't described, which is like the geometric model actually. And they're both here with different degrees of precision. And here then is the same thing for 38 UK dragonfly species. So to conclude, this is ongoing work. There's emergent papers as we speak being written. I haven't talked about dynamic occupancy. This is a vitally important type of model, and it allows us to estimate species movement, for example, the kind of thing that would underlie the kind of movie that we were talking about earlier based on individuals. Okay. Alex Diana at Kent has produced a much faster Bayesian approach for occupancy data. We have an R package for the generalized abundance index models. And James Clark, a research student at Kent, is experimenting with varying spatial scales, different types of land cover covariates, for example, that I didn't talk about. We have a probabilistic red listing method and all sorts of recommendations to do with design, accommodating possible biases due to where the data come from, and recommendations as to what sort of data you can apply these methods to. Perhaps you don't have enough data, for example. So the last slide is to thank everyone that runs these schemes, in particular the UK BMS, but also the people, the citizen scientists who send in their data from the field, as well as the Rothamsted Insect Survey, the moth recording scheme, and so on. And this is the last slide, and it covers, it describes papers covering much of what I've mentioned, and so I'll stop there.
Thank you very much, Byron, for a very interesting talk. We have time very quickly for maybe one or two quick questions. The first is from Mylan Joshi, who asks, is there a trade-off between the speed and quality of different models, or do the alternatives to GAMs actually fit and predict better as well?
I didn't quite catch that. A trade-off between what?
Between the speed and quality of the different models. So do the GAMs also fit better, or do the new models that you develop fit as better as well as being faster?
That's an interesting question. The GAMs they were designed to fill in gaps in the data, and there are plenty of gaps. But the original GAMs actually discarded data because of these gaps, and that's really incredibly wasteful. The more recent GAMs didn't have that feature, but were incredibly slow. But to get to the point of the question, we don't really sacrifice anything when we have these faster methods, and that's why they are now being happily used. They wouldn't be used if people thought that they were second best but fast.
Great, thank you. And the second question is from Ian Jolliffe, who asks, are inter-observer variations in species spotting and identification abilities taken into account? And if not, do they need to be?
Well, it's a very good question. I mean, I'm old, and I prefer to be in the field with, say, my wife, who's somewhat younger than I am. But the ideal is to have one of my children there, simply because they see things. In terms of birds, for example, everybody hears things that I can't hear. This is well known, and there's been quite a lot of recent work actually coming out of, for example, people involving authors from the British Trust for Ornithology, Alison Johnson, for instance. Yes, in demonstrating the importance of knowing the demographics of the people producing the data, so that can be accounted for. So when you have citizen science data of the basic kind, like people just saying they've seen a species, that kind of information isn't present. Because people don't say, you know, I'm 45, I've just seen something. They just say what they've seen. So that's a good point. And if you can, you should take account of that kind of data. Some data sets naturally have that sort of data, others don't.
Okay, thank you, Byron. Unfortunately, we've run out of time for today and we'll have to leave it there. I'm sure if there are any additional questions, then I'm sure Byron will be happy to answer them further. Thank you very much for joining, and I hope you enjoy the rest of your conference. Thank you very much.